Okay, a couple of reminders as we get started. Your phone needs to be away. Chapter 15 homework is a bonus homework. That means if you've turned in every other homework so far, it's a bonus for you, extra credits. If you've not turned in every other homework, missed another homework, that means you can make up for that missed homework. Chapter 11, 12, and your 13, 14 homeworks are in your folders in the lab. If you turn them in on time, I have one or two that got turned in at a different time that I found in my binder this morning, so those I'll try to get back in today too. Your test is on Wednesday on chapters 11, 12, 13, 14, and 15. That's 5 chapters. Hopefully you've already started studying. We're going to do a little bit of review today. The activities that we do today, I did post on Moodle, or I will post them. I don't think I will post them on Moodle, and I'm going to put them in the lab so you can practice from them too. These are not, this does not mean these are the only things you need to know from these chapters. These are just things that you can do to help you study for these chapters. In terms of length, it's going to be similar, but again, it's still a lot of material. So break it down, set aside tonight, do 11 and 12, tomorrow 13, 14, 15. I don't know how you want to do it. Hopefully, we've already started. I was thinking about you and your studying and me and my teaching, and I don't know, I'm trying different things for you to figure out different ways that I can make the material better for you. That also might mean that you need to try some different things. If what you are doing isn't working for you, you need to change what you're doing. If you're not seeing any differences in your results, you need to change your process. So think about that as you're studying. If you've been studying the same way for the first 3 tests and nothing is changing for you, you need to try something different. Because you're going to get the same results if you keep trying the same thing. Okay, so think about that as you are preparing, and we're going to finish chapter 15 today. We have 2 slides to go, I think, and then we will do some review together. So, chapter 15 we left off with on Friday, and we were talking about X-linked inheritance, and you worked on a pedigree. We were looking at hemophilia and the way hemophilia is passed on with X-linked inheritance. Hi, everyone coming. There's some room up here too. Sorry, having a big class. There's some room always in the front, but you probably don't want to be in front. Yeah. X chromosome in humans and also in animals. And so this is an example of what happens when you have X-linked, when you have a gene that's on the X chromosome, and you're talking about the inheritance pattern in, especially in females. So we're looking at X chromosomes, and we're looking at the example of cat for color. Anyone in here? How many people are cat people? Dog people? I hate all the animals people. We won't throw anything at you, okay? Okay, so we're looking at cats because cats have a really interesting fur color inheritance pattern. And so we're going to talk about my example too. So on an X chromosome, we're talking about a female cat here. Females have 2 X chromosomes. And this female cat is carrying 1 X chromosome that has the allele for orange fur, and 1 X chromosome that has the allele for black fur. So they have 2 different color alleles. What normally happens when we have 2 alleles, if it's a trait that has complete dominance? One will cover up the other one, right? So you might think black is probably dominant, orange, and the cat's going to be all black. Well, that is not true. So what happens when you have 2 X chromosomes? As one of those X chromosomes is inactivated. During embryonic development. That Randy's there. So remember, we talked about the X chromosome is way larger than the Y chromosome. It's carrying a lot more genes, a lot more information. So you don't really need both of them to be functioning. So during embryonic development, 1 X chromosome is inactivated, and that inactivation is random. So that random inactivation means that in this cat, sometimes the orange allele chromosome is inactivated, only the black allele is going to be expressed. And sometimes the black allele X chromosome is inactivated, and only the orange allele chromosome is going to be expressed. So what you see in this cat is in parts of her body, the orange allele is inactivated. And in some parts of her body, the black one is inactivated. So we have in some parts of her body, the orange allele is expressed, and in some parts of the body, the black allele is expressed. So that inactive chromosome becomes what we call a Barr body. And it's named after the guy who figured out that this happens. It's also still active in the ovaries. Why would that be really important that both X chromosomes are active in the ovaries, even if they're not everywhere else in the body? Right, and you can't just inactivate one X chromosome, because that would mean half of the eggs that would be produced would be not viable, because they would be missing a chromosome. But that means there's gonna be, again, a lot more variety in the offspring, that genetic diversity that occurs from meiosis. So I said this was my example. In September. In September, I was going into my garage and we've had this feral cat running around our neighborhood all summer. We've been seeing her. She's a very good hunter. We'll just say that. We watched her kill multiple chipmunks in 10 minutes. We started calling her Vicious. So we had this cat named Vicious, and so she didn't like us. She would stay away from us. And in September, we were going to the car to go to school, and the cat came around the corner of the garage and like started meowing at me. And I was like, what's up with you? Like, and I was like, well, we gotta go to school. So we go to the car and she runs in the garage and jumps in the fish tank, because we have an empty fish tank in our garage like everybody. And I was like, are you peeing in there? Because it's really gross and I'm gonna be mad at you, but I gotta go. And I looked and I was like, oh, she's kittens. And so she had her kittens in the fish tank in our garage for a week, and then she got mad at us because we kept going in and out of the garage. And so she took them away, and I was like, whatever, they're gone. And then Columbus Day, I went downstairs to do laundry, and I heard this weird noise in the window well, and I look, and there she is sitting in the window well with her kittens. And I was like, oh, hello, I see that you have come back with the kittens. We've been feeding her. And so she moved into the window well with the kittens, and we could watch them, and she still hated us. And about a week later, my husband was trying to like get her to be friendly, like feeding her. And we eventually got to the point where we were letting them in and out of the house. And one of the kittens tried to go out of the house, and she like picked it up and like brought it back in the house. I was like, okay, I guess you're staying in here for now. So now we have 5 cats in our house. We have 0 cats in our house. We have no plans to have cats in our house. Now we have 5 cats in our house, but the mom is a tortie. So she's a classic example of X chromosome inactivation. So I think that she came to us so that we would have this example of X chromosome inactivation, so I could talk about it. So we've been wondering about the dad because we don't know anything about the dad. We saw this gigantic orange cat wandering around the neighborhood, and there's the 4 babies. So she has 4 kids, 4 kittens, a black one that's fully black with a little bit of white, 2 orange tabbies, and 1 tortie. So we know that the tortie was a girl because it has to be a girl to express that genetic phenotype. So my poor children, I've been hearing about this for months. We've been talking about this, how she has to be a girl. So the little one is a girl, she's the wrench. And these 2 orange ones are male because, why? How do I know that they're male? Besides I checked, but how do I know that they're male? I know that the mom had this and this for hers. So she passed on, if we think of the coat color as, we're going to call it, black is dominant over orange, but when the mom goes through meiosis, that means her eggs are going to either have the big B or the little b. So that means when she passes on the gene, she's either passing on the black gene or the orange gene. And what is dad giving?
Where is it carried? This gene is on what chromosome? The X chromosome. So what's dad giving? A Y, which doesn't have a gene on it. So that means that the babies are either, if they're male, are either XB, big BY, or X little BY. So I know that these two orange ones are X little BY because they got the orange gene from mom and no gene for color from dad. So it didn't really matter if the dad was orange. And I looked more closely yesterday and discovered that the black cat, which I had assumed was also male, was actually female. So I haven't figured this out yet what the dad might be. I think that the orange cat might not be the dad. After all this months of thinking he was the dad. But the female, the black female has to be either this or this. So it could be still the orange dad if the dad passed on this orange gene. So it's like a perfect little Punnett square. I have a little Punnett square of kittens at home. If anyone needs a kitten, let me know. I think I might have one that I still need to get rid of. My family can't decide which one we're keeping, but so that's also true of calico cats. Calico cats are the same thing with the X inactivation. They have the black and the orange, but there's another gene that controls the white color fur that they get the white color fur from because calico cats are tricolored cats. So X inactivation in cats. The last thing that we need to talk about is non-disjunction. So non-disjunction is a process that can happen in meiosis. And this results in an altered chromosome number or an altered chromosome structure sometimes. So how many chromosomes does a human have generally? 46. So if non-disjunction happens, it can either add a chromosome or take a chromosome away. So a normal human karyotype is going to have 46 chromosomes. So if we're talking about non-disjunction in humans, you can have 46 plus or minus 1 chromosomes from non-disjunction. So if this happens because there's some kind of error that takes place in meiosis with this meiotic spindles, where they don't pull them apart properly. In what phase are they pulled apart? Anaphase. So if this doesn't happen properly in anaphase and the chromosomes don't separate, we can end up with gametes that have an abnormal number of chromosomes, and that's called aneuploidy. Most of the time when this happens, the cell is not viable. And if you have an abnormal number of chromosomes, an abnormal gamete, even when it's combined with a normal gamete, usually does not result in what we would call a viable offspring. Having too much DNA is problematic and not having enough DNA is problematic. So it doesn't normally result in a offspring that will develop, an embryo that will develop into a baby that is born. But there are a few cases where on certain chromosomes, you can have a viable offspring. Anyone know the most common example of Down syndrome, which is also known as trisomy 21? Trisomy 21, meaning there are 3 bodies, 3 chromosomes present for chromosome 21 instead of 2. So instead of a pair, you end up with 3 chromosomes there. There are a few other ones that can also occur with the sex chromosomes. Some offspring have XXY, so 2 X chromosomes and a Y chromosome. Which develops into Klinefelter syndrome. And then there's also XXX, so having 3 X chromosomes instead of 2, which is called trisomy X. So Down syndrome has an occurrence rate of about 1 in 830 kids. Klinefelter's is 1 in 650, and trisomy X is 1 in 1,000. These occur, all 3 of these occur because of a non-disjunction in meiosis 1. What is the main point of meiosis 1? What is meiosis 1 separating? The homologous pairs. So in meiosis 1, if we're talking about non-disjunction in meiosis 1, we're talking about in metaphase when our chromosomes line up. Generally, the spindle fibers are going to pull apart the homologous pairs. But in this case, what we're talking about is instead of them pulling them apart, one of them doesn't get pulled apart. So when we continue to the next phase and the sister chromatids are pulled apart, this is curved and they're not going to stay right. We're not going to pull apart all the sister chromatids because the homologous pair didn't separate. So this cell would get a normal number of chromosomes. This cell has an extra one. This is normal, and this one is missing one. So if this egg or sperm fertilizes a normal egg or sperm, you would have that extra chromosome present. The only instance of missing a chromosome, where if that cell that only had one chromosome was fertilized, the only instance of a viable offspring that we know of is when one X chromosome is missing. So you only get one X chromosome. Why does it make sense that you could survive with only one X chromosome? What did we just talk about with the other chromosome? Right, one of them gets inactivated anyway. So you're not missing critical information there because one X chromosome is inactivated. It does have implications because that other X chromosome is active in the ovary, which then obviously it couldn't be because it's not there. But you can produce a viable offspring that has XO, and I wrote all over this. So this is just showing you what this looks like. Again, if non-disjunction happens in meiosis 1, you end up with these extra chromosomes. So this cell has one extra, this cell has one extra, this cell has one. Sorry, I showed it wrong there. This cell has one, is missing one, and this cell is missing one. You can also have non-disjunction in meiosis 2. So if meiosis 1 proceeds normally and the homologous pairs separate normally, but non-disjunction happens here where the sister chromatids don't separate. Oops. You end up with this cell, which has, why is my thing not writing now? Which has an extra one. This doesn't want to write. This cell, which has is missing one, and then 2 normal cells. So you have, again, an extra one, a missing one, and 2 normal cells that are formed. Trying to see if there's anything else I want to make sure I say about that. Okay. There are some instances where organisms normally have more than one set of chromosomes. That's called polyploidy. You can have a triploid egg. I can't write any of this. Sorry. Why is it not? I don't know what it is doing to me here. You can have a triploid egg where there's non-disjunction in all the chromosomes, or tetraploid egg where there's non-disjunction and it combines with another one with non-disjunction. This is actually pretty normal in plants, and we'll talk about that. You'll talk about that in Gen Bio 3 if you're a biology major. But bananas are triploid normally, and wheat is hexaploid, it has 6N, and strawberries are octaploid. They have 8N. They have 8 sets of chromosomes in their cells. Okay. That's the end of chapter 15 for us. So that is the end of what's going to be on the test. So we're going to do some review stuff.
How many people did not get a number? Who needs a number? Anybody else need a number? So I put some, I have 3 sets of diagrams practicing. There's some information about chapter 11. This is a chapter 12 and 13 review. And this is a chapter 14 and 15 review. As I said, after class today, I will take these to the lab. So you can keep working on them. Or come back and practice them again if you want. What we're gonna do is divide into groups. I'm mixing you up a little bit. So we have numbers 1 through 12. So ones will meet down here, grab one envelope, do that, and then come back and trade it out. Okay, so if you have a 1, you're going to move, get up and move and come down here. 2 is over here. Send one person to get an envelope. We should have 3 to 5 people in every group. 1s, 2s. Somebody from 3s come grab an envelope and 3s will meet over here. 4s. Okay, so come get an envelope and then find a spot to go. Pick one, because then I can go anywhere. Okay, 5s and 6s, come on now. I think I don't know other people gonna. Find a spot and make yourself comfortable. You guys. Grab one. One, it doesn't matter which one, and you're fine to go. That's it. They were fine when they weren't. Anyone you want, pick one and then do it and you are done with it, bring it back and switch for another one.
So is it like one per group?
One.
She did.
Okay, if you're at 5, hold up 5 fingers. Sarah's looking for you. There's only 2 5s.
Okay.
You're 5? Or you're 3? Okay, 9s, 10s. Come find your people. Are there any other 8s?
Yeah.
Let's
go 8. You guys want to?
Yes.
You want to grab? No, I don't. Okay, no, that's why it's lazy. Oh, the people in the tag. Oh, it's gonna. Yes, yes. Yeah, I don't know. All right.
Different colored stars. I'll print them off. Might be. How do you know this is something like? So you're crossing over. After my. So we're starting to my houses, we're starting. Six chromosomes are lining up.
Oh. That's insane.
Oh.
I kind of zone out. But once it's zoned out. Technically, that's. Like here. That's really nice. Because this cell here and then this cell. Feeling like your group is too big and you wanna let your sister chromatin, you can come grab another envelope if you want. What kind of get a better Hey, go study now. Okay, yeah, just remind me when you know.
And like if we have homework or stuff, I'll just.
I don't even know what. I can't write it first. And then I'll write. Even though like, I'm pretty sure it sounds.
I think I like drained my vagina.
Like, because I tried to memorize like.
Although it's definitely way better than my sweating, like I was doing my training, that's why I was, you know.
I would be more concerned with
How about the
general
writing?
concept of signal.
They're still a little banged up,
signal
but
transduction,
they're
stuff
also
like
a lot
that.
better than they were yesterday, so.
I don't know if
Hooking another, good
Yes.
news, the.
I'm working on it,
days
I
should
think-
be doing good.
Yeah. For those, it would be more likely to be.
Yeah.
I don't necessarily want you to get weighed down in all the details of everything, but understanding the basic idea
The
of
concept of.
inception, transduction. I
Okay.
mean, I think you should be able to recognize all of them, right, and you, like, you should be able to say, oh, GPCR, I know I'm talking about.
Yeah,
Some kind of cellular communication, and I'm not talking about mitosis, but
yeah,
yeah, like all the details of everything in mind. super interesting.
okay, sounds good, yeah, because I start flying all the time, like, okay,
No, it's
I
a
won't be
lot,
able to, yeah.
no, it's a lot, so, I mean, I've also. The test is the same length,
Mhm.
I'm gonna divide between 5 chapters instead
Yeah.
of 2 or 3, so obviously there's gonna be fewer questions on each chapter
Mhm.
anyway.
Yeah,
So if we're talking about 25 multiple choice, we're talking about 5 multiple choice, 5 or
For
6
each.
multiple choice for each
Mhm.
thing, and the short answer should be each chapter.
okay, perfect, thank you.
You're welcome.
Question.
Yeah.
This is irrelevant to the subject, I was curious. So for the X activation, so you know how it's like on the cat, it's patches of black and patches
Yes.
of brown?
It's true for other things too.
Yeah, so is that just because like the X gets inactivated, it's like the cells around it also get activated when it divides? Or is it, that's just the reason?
Yeah, and so, but I mean, it's true, it's really noticeable in cat because that's so, it's true in humans and that's like. Various axes are inactivated, so it's a chimera, where you're like extracting different genes, of the same, the same genes. So the human are chimeras, so they go same colors, you know,
Yeah,
expressed, chromosomes, so we don't look like Calico Cat.
yeah, that'd be kind of funny.
Cool, but maybe not all the time cool.
Yeah, I was just wondering, because that was weird.
Why